Hello everyone, and welcome to today's live broadcast, Protect Your Cells by Proper Pipetting, How Liquid Handling Influences Your Cell Culture Work, presented by Dr. Hanae A, an application specialist focusing on liquid handling at Eppendorf AG, and Dr. Jessica Wagner, an application specialist focusing on cell handling at Eppendorf AG. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Eppendorf. Eppendorf is a leading life science company that develops and sells instruments, consumables, and services for liquid, sample, and cell handling in laboratories worldwide. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that green Q&A button located at the lower left of your presentation window, type your question into the box that appears on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, notice you're viewing the presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower right. If you have any trouble seeing or hearing the presentation, click on the support button at the top right of the presentation window or use that Q&A button to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Click on the button in the bottom left-hand corner and follow the process for obtaining your credits. I'd now like to introduce our presenter, Dr. Hanae A. Henke and Dr. Jessica Wagner. Dr. Henke is an application specialist focusing on liquid handling, specialized in positive displacement dispenser and pipettes at Eppendorf Headquarters in Hamburg, Germany. She joined Eppendorf after finishing her PhD in medical microbiology on the topic of S. epidermidis hip joint infections at the University Hospital in Hamburg. In vitro macrophage studies were part of the PhD research. She focused on molecular biology and genetics early during her studies of biology at the University of Hamburg and deepened that knowledge in food microbiology laboratories and at Harvard Medical School in the group of Professor Roberto Kolter. Dr. Jessica Wagner is an application specialist focusing on cell handling at Eppendorf Headquarters in Hamburg, Germany. Entering a cell culture laboratory as a graduate student for the first time in 2006, she came to Eppendorf with 5 years of experience in cell culture. After studying biology at the universities of Marburg and Düsseldorf, she investigated apoptosis mechanisms in different cell lines for her diploma thesis. Following 1 year at research associate working with primary cells, she started to work toward her cell of PhD with focus on reproductive biology at the medical school Essen. She joined Eppendorf in April 2012, shortly after completing her PhD in biology. Please join me now in welcoming these professionals, Dr. Hanae Henke and Dr. Jessica Wagner. Welcome, ladies.
Thank you very much for the kind introduction, Susie. So good morning, good afternoon, and good evening, everyone. Thank you for joining us to this webinar, Protect Your Cells with Proper Pipetting. We will discuss how liquid handling influences your cell culture work. My name is Hanae Henke, and I will discuss all the pipetting relevant topics, and then my colleague Jessica Wagner will go through all cell culture relevant topics. The webinar covers 4 main topics. First, there will be an introduction into pipetting fundamentals. Then, contamination prevention will be discussed before we get into detail on pipetting in cell culture. And in the end, you will get information from cell seeding to cell banking and a summary. So everybody working in a lab is pipetting every single day. And as most things we do every day, we do not think much about it anymore. But when it comes to this critical step in an experiment, everything can be changed and minimal differences in pipetting can decide on a result being precise and satisfying or disappointing. Furthermore, accurate pipetting technique is necessary when publishing data. So everyone following proper pipetting rules can redo the experiment and verify your results. Actually, as we are pipetting so much, It should be one of our major concerns, and then it shows having the highest impact of all techniques on our experiments. On the start of each experiment stands the choice of the correct pipetting tool. So let us have a look on the different devices one may use. The different pipette forms vary in their handling, precision, and throughput. The precision ranges from manual over electronic up to automated systems. But what is the reason for that? In manual pipettes, the technique and speed is under user control, while in electronic pipettes, the technique is under user control and the speed is under device control. For automated systems, both technique and speed are under device control and therefore most precise. So each pipette tool is ideal for one application. For example, a pipette helper with serological pipettes for large volumes, like transfer of liquid from cell culture flasks, medium bottles or conical tubes, etc., or, for example, exchange of culture medium. Single and multi-channel pipettes are ideal for applications where the precision is more important, like thawing of stock cultures or subculturing. When it comes to small volumes, less than 1 milliliter, the requested precision and accuracy are much higher, and these small volumes are mainly used in cell seeding, addition of compound and assay reagents, as well as when working with cryovials and microtubes. When using multi-well plates, a dispenser or multi-channel pipette makes sense to add medium and compounds fast and precise. Dispensers can be used from 1 microliter up to 50 milliliter by using the same device and just changing the tip size. So what we see here is that each device has its optimal tip. They all look different and they are all optimized for different applications. Additionally, it is recommended to use the tips manufactured by the same company as the pipette tool, since only then a perfectly working system can be guaranteed. One special challenge in cell culture is pipetting of problematic liquids, like cell culture medium that builds up a lot of foam due to the high protein content. Viscous liquids like DMSO are not only used as cryoprotectants, but also in a lot of cases as solvents, for example for drugs or inhibitors. And due to its viscosity, a large amount of liquid can remain in the tip when pipetting DMSO, which leads to false concentrations and a lot of waste of this chemical. Also, ethanol is quite often used in cell culture labs, either as disinfectant or as solvent for any lyophilized components, but its high vapor pressure makes it difficult to pipette correct volumes since ethanol tends to drip out of the pipette tip after aspirating. To deal with these liquids, we will take a look on the different pipetting mechanisms in the next slides. But first of all, I would like to ask you a poll question. So, which pipetting instruments do you use for cell culture? Please select any that apply. We have 4 answers: manual and/or electronic dispenser, manual and/or electronic air cushion pipettes, electronic pipette controller, or classic pipette controller like a Peleus ball. So please make your choice in the next few seconds, and then we will have a look at the results. So I think everybody made the choice already, and we can have a look at the results now. Okay. So far, we're seeing that nobody answered the question. Usually, okay. We have to wait a few more seconds until we see the results of this poll question. So I'm very curious on which pipetting instruments you are all using for your cell culture applications. Can be 4 different choices. No, most people are using manual pipettes, manual air cushioning pipettes. Okay, so here we get the results. So 64% answered that they're using manual pipettes or dispensers. 23% answered that they're using manual and/or electronic air cushion pipettes only. So 6 answered that they're using mainly electronic pipette controller, and 3.6% answered that they're using mainly the classic pipette controller. So this is really interesting. Thank you very much for your answers. Let us have a look at the comparison of air cushion and positive displacement. So what are the different mechanisms? So some of you are using air cushion pipettes, which are the classic pipettes, and some are using already the dispensers. The classic pipettes are air cushion pipettes. The name derives from the air inside the pipette that is between the actual sample liquid and the instrument's piston. When the air is pushed out by pressing the top button, according to the amount of air, the liquid is soaked into the pipette tip when the top button is slowly released in the liquid. As you can see, the liquid has direct contact with the air inside the pipette, and therefore the formation of aerosols can lead to contaminations inside the pipette. This is what you see as the pink stripes inside this pipette. We will talk about aerosol formation in a few minutes because this is also very interesting and a very big topic, especially in cell culture. But this air cushion system is widely distributed and only suitable for aqueous solutions, since these pipettes are calibrated to water. Problematic liquids with a high density like DMSO or high vapor pressure like ethanol cannot be pipetted accurate without a special technique, like reverse pipetting or a readjustment of the pipette. Less sensible are positive displacement pipettes, also known as stepper or dispenser. This is what you see in the upper right graphic now. Here the liquid is soaked into a tip with an integrated piston. There is direct contact between the liquid and the piston, so there is no air cushion. The risk of contaminating the pipette from the inside is eliminated thereby. And this system allows the transfer of all liquids, no matter if they have a high density or high vapor pressure. Dispensers are more precise and more accurate than air cushion pipettes when using problematic liquids. You can imagine them working just like a syringe, where the piston presses out the liquid. The tip of the dispenser is filled once, and then the liquid is dispensed in multiple steps. It is especially suitable for cell culture when working with plates, so to work faster and more precise than with air cushion pipettes. If you want to know more about pipettes and pipette calibration, we also offer liquid handling seminars in many countries. You can just contact your local salesperson or visit the homepage to learn more about special pipette calibration seminars. Or you can also visit one of our pipette calibration webinars to learn more about it. So now I will hand over to Jessica for the contamination prevention best practice.
Thank you, Hanae, and welcome also from my side. In this next chapter, I would like to talk about some general aspects of reducing the risk of contaminating your cells. But before I start, here comes our next poll question, and we ask you to participate by clicking. So the question is, have you ever had any problems with contamination in your cell culture lab? And the answers you can click on are never, rarely, sometimes, or on a regular basis. So I'm repeating the question once more. Have you ever had any problems with contamination in your cell culture lab? Never, rarely, sometimes, or on a regular basis. Okay, and we will wait a couple of seconds until the answers are coming in.
So we don't see the results right now. I'm sorry, I think we have some technical problems here. Okay, so they are coming in. We have 55% of you who say they rarely see a contamination. 33% of you have a contamination from time to time, and only 6.5% of you never had any issues with contamination in their lab. So we see that it is still a common problem in cell culture labs. Okay, so contamination is maybe the biggest challenge when working with eukaryotic cells. In general, we find two major types of contaminants, the microbial ones and the eukaryotic. The microbial contaminations are fungi and yeast, bacteria, and a special type of bacteria, mycoplasma. The other ones, the eukaryotic, are not necessarily so well known and are cross-contaminations of other eukaryotic cells in cell lines. This phenomenon was first described in 1967, and to date it is estimated that 15 to 30% of all cell lines used in labs worldwide are either cross-contaminated or misidentified. No matter what type of contamination we are talking about, I think we can all agree about the consequences. Loss of time, since you have to repeat experiments. Increase in cost, time is money, and you need more material. Loss of reproducibility, you might have completely different responses when you work with contaminated or non-contaminated cells. And the last point might be the most horrific, the loss of significance. If you want to find a new therapeutic treatment of breast cancer, but you work with melanoma cells without knowing it, the results of your work are useless. So taken together, avoiding contamination is much easier than dealing with it. There are so many possibilities of getting contamination, so prevention is always the logical step. Maintaining a sterile environment has first priority in a cell culture lab. Instead of sterile, I should better call it germ-reduced environment, because this is the actual truth. For example, the inside of an incubator is never sterile. Every time the door is opened, germs can enter the incubator, plus the atmosphere inside the chamber is disturbed. Therefore, door openings should be reduced in duration and frequency as much as possible. Preventive hygiene plays a big role in preventing contamination. That means regular cleaning of lab equipment like incubators, water baths, biosafety cabinets is a must. Also, personal protective equipment is very important. You should have a dedicated lab coat that you only wear inside the cell culture lab. Every other type of personal protective equipment, like for example lab shoes, further reduce the risk of contamination. Your workspace in the laminar flow hood is the place of action and the place where most mistakes can happen. I will come back to that point in more detail later. All preventive hygiene and personal protective equipment and organization is useless when you don't take care of your aseptic technique while handling cells, reagents, and last but not least, pipettes. The person who is working in the cell culture lab is maybe the biggest source of contamination. Your hands are the major source of germ transfer when handling cells and sterile liquids. It may be trivial to specifically mention, but when entering the cell culture lab, the first thing to do should be washing your hands. Disinfecting your hands before putting on gloves further reduces the contamination risk. Another advice is to use hand cream regularly because healthy skin has fewer germs than dry skin. When you put on gloves, disinfect them sufficiently. That means let them dry before you start working. And disinfect them not only once, but repeatedly. For example, when you adjust your glasses, never do it without subsequent hand disinfection. You can also tuck the sleeves of your lab coats in the gloves. I already mentioned the importance of having a lab coat that is reserved for the cell culture lab. Same applies, of course, for any other type of equipment. Especially pipettes and tip boxes are easy to carry from one lab to the other, which should be strictly forbidden. A good tip is to simply put a sticker on each pipette and label it with cell culture, so that everyone knows that this pipette should be kept inside the cell culture lab. Disinfect the pipettes each time before you place them in the hood.
Do it thoroughly by wiping with a disinfectant to allow a complete wetting of the surface. If you choose a new pipette for cell culture, consider one that is easy to clean with a smooth surface that does not have many rough edges or grooves. You can also disassemble the pipettes to clean them from the inside if necessary. Autoclavable pipettes give extra safety. When you are pipetting, try to avoid moving your hands or arms over open vessels. At the beginning of this chapter, I mentioned the importance of having an organized working place in the biosafety cabinet. As this is the area where you open the dishes, plates, or flask with the cells, there are some things to be kept in mind to avoid contamination. You should always work in the middle third of the workspace, not too much in the front, not too much in the back, because there you disturb the laminar airflow in the cabinet. Another advice is to not block the air grills in the front, the back, or at the sides with research notes, discarded plastic wrappers, pipetting devices, and so on. Disinfect the working area in the hood completely and let it dry before every use. Disinfect every piece of equipment that you bring in. Every item that is in there disturbs the laminar airflow that keeps the germs outside. So don't overcrowd your workspace with items that you don't need for that particular working step. Any droplets or spills need to be cleaned immediately. When working at the hood, it is a good advice to have one clean side with culture media, pipettes, and new consumables. The other side is reserved for liquid waste, used tips, and so on. And with this, I will hand over to Hanae A again, who will proceed with the next chapter, pipetting techniques in cell culture.
Thank you, Jessica. And now let us have a look on what is most important in cell culture experiments. So there are three major important things. The first one is the protection. So in the following slides, we will use protection as a term to describe that, of course, all steps have to be performed under sterile conditions, but some methods are safer than others. For example, distance to cells or reagents. Serological pipettes with a distance of about 30 centimeters to potentially non-sterile hands and electronic pipetting controllers or dispensers have a bigger safety than small dispensers with only approximately 10 centimeters distance to the sample, compared to a pipette tip, which is only 4 centimeters of a distance. So a serological pipette is usually safer than the others, even when special precautions are taken simply by the larger distance. The second topic is the precision. This is the term we use in all slides, but we actually refer to the validation terms precision and accuracy. With this word, we describe it's an explanation that would go beyond the scope of this presentation to go through all the precision and accuracy validation needs. And this is a part of the pipette clinics, but nonetheless, precision will be a big topic in the next few slides. And the third one is time and cost, which is, for example, the pipetting of a 96-well plate with a single channel versus a multi-channel pipette. And this is a good comparison of speed and therefore also the cost per sample. So I would like to start with the main topic of protection, which is aerosols. What are aerosols at all? So aerosols are formed always when a liquid is transferred, especially when using warm liquids like cell culture medium at 37 degrees Celsius. In room temperature conditions, a slight evaporation leads to very small aerosol drops that can contaminate the inside of the pipette and with this lead to cross-contamination of the sample. As you see in the illustrated pipette tip, an effective barrier against aerosol formation is a filter tip. It stops small aerosols and contaminants such as bacteria or virus to get into the pipette. However, using a dispenser with a fresh tip every time is 100% contamination-free, and the more valuable choice when it comes to very sensitive and expensive samples. Let's have a closer look on filter tips. When using filter tips, remember to use a two-layered filter. Its first layer retains drops, splashes, and large particles like bacteria. The second layer retains small aerosols, biomolecules like DNA and RNA, as well as virus particles that could contaminate the pipette cone and cross-contaminate your samples. But not all aerosols can be retained by a two-layered filter tip. There is still a chance for very small aerosols to successfully pass the filter. Therefore, using a positive displacement system is still more secure. We have some more tricks to avoid aerosol formation, which are gentle mixing by rolling rather than vigorous shaking, or pipetting directly into a liquid or onto a surface to avoid bubbling and splashing. Therefore, a fresh tip for every pipetting event is absolutely demanding. The third one is to place containers very close to each other when transferring liquids between them to avoid drops that fall and may splash into other tubes. And slowly remove tube caps and stoppers. So let us go over to the second big topic, which is precision. What did we learn about precision in pipetting so far? One widely used tool is an electronic pipette helper. But keep in mind that the liquid volume is mainly defined by the person using the electronic pipette helper. The meniscus is seen by the person pipetting and stopped when the volume on the graduation is reached. This is a very personal system, but least precise. A more precise option is a manual pipette. These are suitable for aqueous solutions, but still the technique and speed fall to the user. With electronic pipettes, the speed is device controlled and only the technique is the precision limiting user-dependent factor. The highest precision with all liquids is achieved by using Dispenser. The device dispenses the same amount of liquid on each stroke, either manually or electronic. So also here, electronic devices offer more security because also the speed is under device control. But let us have a closer look on the tools. For large volumes where precision is not so important, an electronic pipette helper is sufficient. For example, when exchanging culture medium. When using flasks, it might also be of advantage to use serological pipettes and a pipette helper. The second option is much more precise, so using pipettes with an elongated tip or a dispenser with long tips avoids contacting the inside of the tube or flask. However, you have to decide whether the protection of your sample via large distance by serological pipettes or a more precise pipetting result by using pipettes or dispensers is essential for your application. So you always have to make the choice between precision and protection when doing your experiments. So you see in this picture that the pipette cone reaches into the tube, which may lead to contamination on the outside of the pipette cone and then in your next sample. So choosing the right vessel according to the application is also essential. The pipette tool used is as important as the whole experiment setup. If you're using a manual pipette, the tube used should not be too high that you touch the inside of the tube, as we just saw in the picture. The 5 milliliter tube is safer to be used with a pipette instead of the 15 milliliter tube or flask. Using a 15 milliliter tube with a dispenser and the according tip is a safe option. You do not get the device in contact with the inner side of the vessel and protect your sample as well as the device. So you see that the whole experiment setup is always important. Each single step must be thought through. So now when pipetting large volumes, you have to think about one very important thing. When pipetting upright over open flasks, contaminations may fall directly into the open flask. So remember to always use a 45-degree angle when pipetting out or into vessels using a pipette controller when doing large volumes. Now, when pipetting small volumes, precision is most important for your assay and the addition of your compounds. Therefore, a manual or electronic pipette or a dispenser are most suitable. But when using these, you can see that there are some very important things to consider. So on these illustrations, you can see two major things to remember during the aspiration of liquid. First of all, the immersion depth, meaning how deep you put the pipette tip into the liquid, and second, the angle you're using when holding the pipette tip. Diving in too deep raises the surplus liquid volume inside the pipette tip. You aspirate too much liquid. Additionally, holding the pipette at an angle of 30 to 40 degrees, which you see in the third picture on the far right, raises the surplus liquid volume even more, and you result in an imprecise pipetting result. So, ideally, holding the pipette tip upright vertical at an immersion depth as shallow as possible leads to the most accurate liquid aspiration possible. Keep in mind that for liquid volumes below 20 microliters, a very low immersion depth is recommended, like only 1 millimeter. While for liquid volumes above 20 microliters, you might immerse the tip a little deeper, but never more than 5 millimeters. So you realized one big difference. When doing large volumes using a pipette helper, to avoid contamination, you should use a 45-degree angle when aspirating and dispensing. But for small volumes, when precision is most important, you should use the upright angle. So there again, you have to make the choice between precision and protection of your sample. So now we will go to some more interaction and have the next poll question. And I'm very interested in, do you use a dispenser for small volumes? And there are three very easy answers. It's yes, no, or I'm not sure how to work with these devices. So we will wait a couple of seconds, and then we can shortly discuss your answers and go through the topic of dispensers and dispensing small volumes using multi-channel pipettes and dispensers. So I think the results will come in now, step by step. So we don't see the results at the moment. It will be just a few more seconds to show up. Okay, so I think we will see the results in a few seconds. I'm just waiting for them to show up. So I just got the message that they are coming in. So I'm very curious to see if you are using dispensers for small volumes or if you're not using them at the moment, or if we should discuss how they work. So, okay, 35% of you answered that they are already using dispensers for small volume. 50% answered that they're not using the dispenser for small volumes at the moment. And 13.9% answered that they're not sure how to work with these devices. So we will discuss on how they work a little later. So it's interesting, nice to see that 35% are already using the devices because they're great for small volumes. And maybe we can convince some of the 50% and the other 13.9% to also give the dispensers a chance for small volumes. But first of all, we will go through dispensing et al., so dispensing with air cushion pipettes, and then the next slide will be about dispensing small volumes using a dispenser. So when dispensing the liquid, a 45-degree angle and securing the fill tip on the wall of the vessel is fine. Yeah? A blowout is absolutely necessary when using manual air cushion pipettes. With dispensers, a blowout is not necessary since each dispensing step delivers the correct liquid volume. So you avoid air bubbles. While dispensing at an upright position, you should avoid dispensing at an upright position to not stress yourself additionally by liquid directly spotted on the cells. So do not move your hand over the plate to avoid contamination. So in this case, it's also a decision between precision and protection, because the 45-degree angle means you have a less precise pipetting result, but it protects your cells to use a 45-degree angle when dispensing. So if you're using a dispenser now, they make the whole pipetting process much easier. And you do not have to think about immersion depth or aspiration angle. Additionally, you can use any liquid with a dispenser, no matter what property the liquid has, like viscosity, density, or vapor pressure. So, as we already learned that dispensers are using tips that look like a syringe, you can aspirate using a syringe in any angle. Because there is no air cushion, so there is no effect, you can use the secure 45-degree angle and still have the best possible precision when using a dispenser. Okay, so you can choose the best option for your cells in protection as well as precision using a dispenser. And using a very, very small dispenser tip, like a 1 microliter tip, using an electronic dispenser with this tip, there you can choose to only aspirate like 5 times. You don't have to fill the tip completely when dispensing small volumes. So one thing why people don't use small volumes with dispensers is because they think they have to fill the complete dispenser tip. That takes about 100 microliter, and that would be a lot of waste. But you don't have to fill the complete tip. With an electronic dispenser, you could only aspirate 10 microliters and then dispense 10 times 1 microliter. So you don't have any waste, and but you have the best precision and the best protection for your cells during your experiments. Okay, so now I will hand over to Jessica again for the next part.
Okay, so in this last chapter, I want to give you a few practical tips on some of the most critical steps in your cell-based experiments or in your cell culture routine. So let's have a closer look at this first experimental step that is so important, cell seeding. The precondition for getting comparable results is to seed out the same number of cells for every negative control, positive control, and for every experimental replicate, of course. The more replicates you include, the better for your statistical analysis. Variations in seeding cell numbers and the formation of air bubbles during seeding will not only increase standard deviations, it will also make your results less reliable. Repeated mixing of the cell suspension during seeding and careful pipetting can be a solution. So let's go more into detail on the next slide. When you fill, for instance, a multi-well plate with cells, it will take some time to fill all the wells. You can use a single-channel pipette and have the cell suspension in a tube, or you can use a multi-channel pipette and have the cell suspension in some type of reservoir. No matter which type of tool you use, the longer the seeding process takes, the higher is the variation in cell number from one well to the other. As you see in this little cartoon here on the right, the cells settle in the tube with time, and this sedimentation goes quite fast, within minutes. Therefore, you need to resuspend or mix the cells to swirl them up in between. Thus, you get an equal number of cells in every well. Saying that, we should keep in mind that cell culture media tend to foam due to the high protein content of supplemented serum. Air bubbles can hinder cell attachment, so you should avoid harsh and fast pipetting to reduce the formation of air bubbles and also to reduce shear forces, which further stresses the cells. I want to end this chapter with, okay, so now let's have a look at how pipetting can affect your assay results. In this graph on the left, you see the results of a cell viability assay. Each column represents one row of a 96-well plate. The aim was to seed an identical number of cells in each well. 2 rows were left out for blanks here. So the cells were seeded with a manual 8-channel pipette from a reservoir. After 24 hours, the number of viable cells was determined using a colorimetric assay. In rows 1 to 5, you see varying cell numbers in the wells and therefore high standard deviations. Here the user did not mix the cell suspension in between pipetting each row, so the cells settled in the reservoir with time. In addition, the pipette was held in different angles and heights during aspiration. In rows 6 to 10 you see the results when mixing the remaining cells before pipetting each row, aspirating at constant height and in upright position. Taken together, these data show that the pipetting technique has a big influence on the result. You can also use a dispenser instead of a multi-channel pipette for applying assay reagents or seeding cells. With a dispenser, immersion depth and pipetting angle do not influence the precision of the instrument. With this next slide, I want to talk about another very important step in cell culture, the cryopreservation of cells. When you want to establish frozen cell stocks of high quality, the stress factors for the cells need to be minimized. Cryoprotectants like DMSO are harmful to the cells in non-frozen media. When cells are freshly thawed, they are very sensitive to shear forces. So taken together, time is a critical factor when freezing and thawing cells. When you freeze the cells, you should be quick from the point where you add the DMSO-containing freezing medium to the cells until you start to cool them down. The cooling procedure itself should then be slow to avoid the formation of ice crystals in the cells. To achieve a cooling rate of 1 degree per minute, use a special freezing container and put the cells into a -80 freezer. You should not store the cells for longer time at -80, but freeze them down in liquid nitrogen or in a -150 freezer. When you thaw the cells, time is again critical because you want to minimize the exposure to DMSO. So thaw the cells as rapidly as possible and dilute them to decrease the concentration of DMSO in the medium. After that, you can either remove the DMSO directly by centrifugation or dilute the DMSO and change the medium the next day. And again, it is important to mention that freshly thawed cells are sensitive to shear force, so too harsh pipetting should be avoided. In general, it is a good advice to not work with too many samples at a time and be organized. For example, label cryotubes and culture vessels first before you start to prepare the cells for freezing and thawing. I want to end this chapter with some general remarks about cell banking, because having high-quality cell stocks is your backup system and the prerequisite for reliable research. The most important point is that you make sure that you don't freeze contaminated cells and that the cell line is really the cell line you think it is. So authentication of cell lines should be performed regularly, especially when you got the cells from another lab and when you use them in long-term or subsequent studies. The cells you are working with should also be tested for mycoplasma regularly. As Hanae A mentioned earlier in this presentation, filter tips give extra safety, so they should be considered at least for such important steps like cell banking. The routine use of antibiotics is not recommended because they can mask a contamination with partially resistant bacteria. And please keep in mind that most antibiotics are not effective against mycoplasma anyway. The two-stage cell banking system with a master and a working stock provides you constantly with early passages from your cell lines. A detailed record of the cell stocks is as important as clear labeling of cryotubes. So to summarize what we were discussing in this webinar, here we have again the 3 aspects that are important when talking about pipetting in cell culture: precision, protection, and time. When a precise volume is needed, for example, when pipetting assay reagents, it is important to hold the pipette upright during aspiration and to use a constant immersion depth. When you empty the tip, rest it on the vessel wall and blow out the liquid completely. When seeding cells, for example, in a multi-well plate, it is very important to mix the cell suspension in between pipetting steps to achieve equal cell numbers and enhance reproducibility of your experiments. But make sure that you avoid the formation of air bubbles. Pipettes are precision instruments. To keep the precision, it is essential to calibrate them on a regular basis. The second aspect we were discussing is protection against contamination. You can reduce the risk of contamination when you hold the pipette in a 45-degree angle so that you don't have your hand directly above the open vessel. For example, when you are feeding or subcultivating the cells, protection is more important than a precise volume. Of course, all pipetting tools used in cell culture should be thoroughly disinfected before each use. It is also good to autoclave pipettes regularly, but please check with the manufacturer first if the pipettes you are using can be autoclaved. Having a dedicated set of pipetting tools that don't leave the cell culture lab and using filter tips, at least for the very important steps like freezing cells, help further to protect the cells against contamination. When handling cells and media in the biosafety cabinet, the workspace should be organized. Don't overcrowd the hood with items that you don't need. Work only with one cell line at a time and separate clean pipetting tools, tips, vessels, and media from liquid waste, used tips, and so on. Last but not least, time can be a critical factor. For example, the time that it takes for filling all wells of a 96-well plate. Using multi-channel pipettes or a dispenser can save you a lot of time. Dispensers are a good choice for problematic liquids like DMSO or cell culture media. Remember how important it is to avoid the formation of air bubbles when seeding the cells. With a positive displacement system like a dispenser, you avoid air bubbles and you can be very quick in filling multiple wells or dishes. In general, you should not work with too many samples at a time to avoid that the cells are too long at room temperature or reduce the exposure to DMSO when freezing or thawing cells. Before we come to our last slide, here comes our last poll question. So please let me know how you rate the content of this webinar. You can choose between very good, good, okay, or poor. Okay.
Thank you so much, Dr. Henke and Dr. Wagener, for your informative presentation. We want to now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window, type your question into the box that appears on the screen, and click that send button. We'll answer as many questions as we have time for in our presentation today. And thank you so much for participating in the polls. We have a great audience today. Here's our first question. This one is for you, Dr. Henke. Are there any cell health consequences when using a dispenser such as the M4 to dispense cells? The second part of that question is, I'm thinking about the small orifice size and high velocity, possibly shear forces, when dispensing using the M4.
Thank you very much for this question. It's a very interesting question, and we hear this many, many times. That's why we tested exactly our multipad, or it's called repeater in the US, the model M4 with cells to see if the orifice has an influence. And we published the data in an application note, which is called Multipad Repeater M4 allows for fast, precise, and sterile liquid transfer in cell culture. So we could actually see that the cells are very fine when using the M4, that they grow well, that the cell count, the cell number is still fine, and that you can absolutely work with the Multipad Repeater M4 and the dispenser tips with cells. You can download this application note from our homepage. I think if you use a search engine on the internet, just typing Multipad Repeater M4 and cell culture, the third or the fourth result is the PDF document of this application note, and there you can read through all the results.
Thank you, Dr. Henke. And what are the differences, Dr. Henke, between manual dispenser and classic pipette?
So we discussed this difference in the beginning. The classic pipettes have an air cushion in between the sample and the piston inside the pipette. So each sample is also dependent on this air inside the pipette, while in comparison, in a manual dispenser, you're using dispenser tips, which have the piston integrated in the tip. And there, the sample is in direct contact to the piston, so there is no air cushion. It's working more like a syringe. While in a classic pipette, you have the liquid only in the tip, which is made of some plastic material. Then there's a lot of air, and then there's a piston inside the pipette. So there are complete different principles of building these pipettes and dispensers.
Thank you, Dr. Henke. And I think you are partially answering this next question. The audience member wants to know, I'm surprised more people use dispensers versus pipettes. Could you explain the advantages of dispensers in more detail?
So the advantages of dispensers, they all come from the principle of how they are built, how they're functioning. Because they have a dispensing tip that's built more like a syringe, you don't have all the problems that come with an air cushion. And an air cushion is usually, it's flexible, so you can compress air and it can be elongated. Everybody knows if air is getting hotter, then it's building up pressure, so it's getting bigger, if you have this picture in your mind. With a dispenser, without the air cushion, you don't have these air effects. So you can use any liquid, also liquids which are hot or very cold or tend to evaporate, and you still have a precise result. So this is the main advantage. You can use any liquid you want and still have a precise and accurate result. Then the second very important advantage, which is very important in the case of cell culture, is that you don't have contamination. Because if evaporation occurs with your liquid, the liquid cannot pass this piston inside the tip, so it cannot contaminate the dispenser. While using a normal pipette, it's possible that aerosols can enter the pipette cone and then cross-contaminate your samples. So these two are the main advantages. There are some more, but I think we want to answer some more questions. But I will be willing to answer this question via email a little in more detail.
Thank you, Dr. Henke. And connecting to that, could you mention again pipetting techniques for viscous liquids?
Okay, so for viscous liquids, I recommend to use reverse pipetting, which means that the blowout volume is also aspirated into the pipette tip as an additional liquid, but it is not dispensed. So if you have a normal two-button pipette where you have an operation button and an ejection button, and when you're using the operation button, you see that there are two different stops. So the first stop is usually used before aspirating the liquid, and the second stop is used for the blowout. When you're doing reverse pipetting, you would press down the operation button down to the second stop, then aspirate the liquid plus the blowout volume, and when dispensing your liquid, you would only go down to the first stop. So there will always be a rest, a remaining liquid in the tip of the viscous liquid, but you have exactly the precise viscous liquid dispensed that you set on the volume.
Thank you. This next question comes, what is the frequency of autoclaving that you typically would recommend, and does repeat autoclaving affect calibration? This is for you, Dr. Henke.
Okay, so repeated autoclaving of our Eppendorf pipettes does not affect the calibration. We autoclave our pipettes here in test cycles, I don't know how many 100 times, but a lot. So I can talk to my colleague, she's a specialist, especially for these pipettes and calibration, to ask in more detail. But it doesn't affect the calibration at all. So you could autoclave your pipette daily if needed, and you can still precisely work with it. The only thing we recommend is after cleaning a pipette with a grease-destroying agent like ethanol, which just destroys greasy things on the piston, then you should re-grease the pipette. But this is not necessary after normal autoclaving, and this does not affect calibration at all.
Thank you so much. This next question is for you, Dr. Wagner. Doesn't directly vertical raise your risk of contamination?
Yeah, I think I think you wanted to ask if it raises the risk of contamination. Yeah, that is actually true. If you have your hand directly over an open tube or over an open vessel, of course there is a higher risk of contamination, because your hands will never be 100% sterile. So even if you have them disinfected, there's always a rest risk or remaining risk. What Hanae A tried to point out is that you have to consider which is the step I'm currently doing. Do I have, for instance, an endpoint analysis? I only add the compound or the assay reagent, and then I analyze the cells directly after it and don't need to incubate them anymore. So you always have to consider between where do I need the precision and where do I need the protection of my cells. When you do the subcultivating routine, you split the cells, you change the medium, you don't need a precise volume. It doesn't matter if it's a few hundred microliter more or less in most cases. So here, of course, you can have the pipette tool in that 45-degree angle that Hanae A recommended. But especially when you have endpoint analysis where you don't need further cultivation of the cells, then I would choose the more precise technique because there it is usually more important.
Thank you so much, Dr. Wagner. This next question is for you, Dr. Henke. One of our audience members are searching the Eppendorf site and would like to know, I'm searching the Eppendorf site for dispensing pipettes, but those keywords only are yielding a bottle version. Can you assist?
Okay, so I think dispensing pipettes is the wrong search term in this case. If you want to search for our dispensers, what I guess is what you mean by dispensing pipettes, I would recommend to directly enter multipad or repeater in the search form on the Eppendorf homepage. There you should be directly brought to our repeater and dispenser versions, where you can read more about the devices and we can watch a video and read some application notes. But I can also send this link via email to the attendees so that they have the right link.
Thank you, Dr. Henke. This next question is for you, Dr. Wagner. I want to know more about protecting the integrity of cell monolayers when doing PBS washes and media changes on 96 well plate cultures.
Yeah, I think there was a similar question from another attendee. Yes, so especially when you work, for instance, under serum-reduced conditions, or you have a cell type that does not adhere so much or so strong on the plastic surface, like, for instance, HEC 293 cells is a good example for that. You have always the risk when you change the media or when you do the washing step that the cells come off the plastic. So of course, you should not directly pipette any type of liquid directly on the monolayer. You should always bend the vessel a little bit so that you can pipette on the walls and not on the growth surface, and then move the vessel around so that the PBS, for instance, covers the complete monolayer. You can also think about if a biological coating is something that could be a choice in your case. For instance, if you coat the surface with polylysin or any other type of coating, then the cells will adhere stronger. But you just have to be very, very gentle to the cells, let's put it that way, and not pipette directly onto the monolayer.
Thank you so much. And Dr. Henke, Dr. Wagner, thank you so much for this wonderful, informative presentation. I also want to thank audience members and remind them that any other questions coming in will be answered and followed up via email. Dr. Wagner, Dr. Henke, would you like to provide the audience with any final comments before we close?
We want to thank everyone for joining this webinar. So we were really happy to have you here and have that many very interesting questions. And we would just like to point out that you can get some more information on cell culture work and how to pipette correctly, some very interesting contents on eppendorf.com/cellexperts, as well as eppendorf.com/pipetting. You can inform about some more webinars and seminars and some more interesting content.
Okay, I think that is it. So thank you all for joining us today.
And thank you both very much. And I also want to thank LabRoots and Eppendorf today for making the educational webcast possible. Before we go, I want to remind audience members to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks so much for joining us. We hope to see you again soon. Goodbye.